I'm Janet Rossand, President and Scientific Director of the Gairdner Foundation. I'm thrilled to welcome you to our second Global Perspectives Panel of 2022. Today, you're going to hear from world-renowned experts on how CRISPR technology is changing health, food systems, and conservation to address food security and climate change worldwide. Our core mission at the Gairdner Foundation is to recognize and reward international excellence in fundamental research that impacts human health, which is done through our 7 annual prestigious Canada Gairdner Awards. But beyond the awards, we believe in the power of open scientific discourse to better engage the public, to help us all understand the problems we face in the world today and work together to find solutions. Throughout the year, Gairdner hosts various events that convene leaders in their respective scientific fields to do just that, and also to really inspire the next generation of researchers. On September the 13th, we're going to host our 3rd Global Perspectives Panel of 2022, which follows up on a series that we have done on the COVID pandemic. This one asks, in the light of where we are today, what did we learn from the pandemic and what comes next? This is going to be followed by our annual Gairdner Science Week, which kicks off on October the 24th and will feature live in-person and webcast symposia, mentoring sessions, panels, and a gala celebration for this year's awardees. We hope you'll join us for some of these events. Stay tuned for updates on our website, gairdner.org, and on social media at Gairdner Awards. And finally, on behalf of Gairdner Foundation, I'd like to thank our presenting sponsor for today's event, TELUS Health. For you, the audience, if you like to enter your thoughts in the Q&A, we will have a discussion session and we will certainly try to get to some of those issues that you raise. But now we're going to hear from our speakers today. And first up, we're going to hear from Dr. Feng Zhang, 2016 Canada Gairdner International Award laureate for his work on developing CRISPR-Cas9 gene editing tools. Dr. Zhang is a core member of the Broad Institute and the McGovern Institute at MIT. Dr. Zhang.
Hi, good morning. It's really a great pleasure to be a part of the symposium, and it's really nice to join all the other panelists and also the Gairdner Foundation. So I am a professor at MIT, and we work on developing molecular technologies for improving human therapeutics. And so today I wanted to tell you about some of the work that we've been working on to continue to advance genetic therapies. So genetic medicine is a really promising platform for developing therapeutics. Unlike small molecule like chemical based therapeutics, genetic medicine is much more modular. So I put on this slide a picture of a rocket delivering a satellite into space. And this is kind of like how genetic medicine works. There are 2 parts to it. There is a vehicle which delivers the medicine into the right cells and also the right tissue in the body. And there's also the payload, which is the therapeutic molecule itself. Over the past decade, there's been a tremendous amount of progress made in the development of payloads, CRISPR-Cas9, and also all of the different ways that people have engineered CRISPR-Cas9 for making changes, for making modulations in cells have really come a long way. But the vehicle for putting CRISPR-Cas9 into different tissues and different cells has lagged. And to date, our ability is largely limited to targeting of the liver, targeting of the eye, and also cells in the blood, which you can remove from a patient's body, modify them outside in a petri dish, and then put back into the patient. So the ways that people have been delivering different payloads like CRISPR-Cas9 really fall into 2 different categories. One is viral-based delivery. This involves the use of viruses and engineering these viruses so that you can, or scientists can remove all of the pathogenic material that makes the virus harmful to human, and only keep the shell of the virus so that you can package new therapeutic molecules like CRISPR-Cas9 into them. These viruses can be made in using human cells, and then the virus can be harvested and then injected into patients to be able to treat a specific disease. And so on this slide, AAV, adenovirus, and lentivirus are different examples of viruses that can be used for making delivery possible. There are drawbacks to these different systems. Some of them are immunogenic, some of them have very limited packaging capacity, and then some of them is very difficult to make at a very large scale so that you can make enough to treat a big organ in the body. Complementary to the viral-based systems are the non-viral systems. And non-viral vectors are based on lipid nanoparticles. And many of the coronavirus vaccines are developed based on this approach, using lipid nanoparticles to package messenger RNA to then deliver things into the body. And so non-viral delivery using LNP has also been shown to be successful for targeting the liver, but for targeting other cells in the body, it remains a very big challenge. But the benefit of LNP is that you can scale this up much more easily. And so we have seen that billions of doses of vaccine have been developed based on this technology. So despite these two different approaches, we still have a lot of unmet needs in the delivery space. We need a system that is flexible for packaging different sizes of therapeutic molecules. We need ones that can robustly distribute throughout the body to target many different tissues. We need ways to be able to target specific cell types and not just non-specifically target everything. And we also need things that can be scalable so that we can make lots and lots of it to be able to treat many people and also large organs in the body. And then lastly, ideally, it can be used multiple times so they can redose a patient to be able to replenish a therapeutic. So the approach that we have been taking is a little different than the virus and also the non-viral based approach. So instead of using lipid nanoparticles or viral vectors, we have been looking at ways to develop things that look like viruses but are not viruses. And so this is derived based on a type of virus called retrovirus. So there are really two main types of viruses. On the left side on the slide are coronavirus and influenza viruses, and these are very much in the news headline today. These viruses infect a person, but once the body clears them, once the body recovers from the infection, these viruses disappear from the body. But retroviruses, on the other hand, integrate themselves into the cells of the patient. And so it leaves a copy of its genome inside of the cell. So it's once a person has been infected with retrovirus, unless those cells are removed, the virus stays with the person for the rest of that person's lifetime. And so needless to say then, retroviruses have existed for millions of years. And so if you look into the genome if you look in the human genome, you can find many examples of retroviruses that have invaded the human genome millions of years ago, or invaded the genome of our evolutionary ancestors, so even before human existed. And many of these genes in the human genome that are derived from retroviral-like systems have homology or have similarities to pieces of retrovirus. And so if you look at one of the domains in these genes, you can see that they carry a piece of the protein that is important for forming the structure of the virus. And so more than 10 years ago, scientists have used computational strategies to look into the human genome to see how many of these virus or retroviral derived genes are there. And they found that there are three major families. There's a family called the sushi family, which are derived from a retroviral system called the sushi retrotransposon. There are also the SCAM family, and then also the paraneoplastic family. And so all these are very similar looking to virus-like proteins, but they're not viruses. They're natural human genes in the human genome. So a few years ago, a couple of research groups led by Jason Shepard and also Travis Thompson published two papers showing that one of these proteins called ARC, which is normally found in the brain, is expressed and it forms virus-like particles. And so that got us to be really excited. And so I thought maybe if ARC can form virus-like structures, probably many of these other genes in the human genome can also form virus-like particles. And more importantly, can we see if they package molecules and can they be used to deliver molecules to other cells? So we began by studying a few of these different proteins. On this slide are 5 of them. And you can see that 4 of them, the 4 on the right, they form virus-like structures. Not all of them do, but most of them have retained this ability. And so this is really interesting. So now we wanted to see if we put these into cells, do they make virus-like particles and do they get released by the cells? And so, so we did an experiment using human cells, and we found that if we put a number of these different virus-like proteins into the human cells, 4 of them in our experiment were able to make these virus structures and release them to the outside of the cells. And one of them was quite robust, and that's a protein called PEG10. So seeing that these proteins can release from cells, we wanted to know, are they packaging anything inside? And what are they packaging? And so by doing some RNA analysis to look at what is included inside of these virus-like structures, we found that they largely package their own messenger RNA. And at first we thought this is very surprising, but after thinking about it for a bit more, this is actually not too surprising, because if the proteins derived from virus-like systems, then these proteins naturally package the genome of these viruses. And so PEG10, the mRNA, is essentially the genome of the PEG10 gene. And so the fact that it packages its own mRNA, in that context, is not that surprising at all. And so based on that, we were then able to figure out what are the pieces, what are the signatures on this messenger RNA that allows the PEG10 molecule to package those RNA. And we found out that it's really the 5' and the 3' UTR that is recognized by these viral proteins. And so this then allowed us to come up with a way to develop a delivery system to package new molecules. For example, the messenger RNA for Cas9 inside the PEG10. And the way the system works is that we introduce into human cells a piece of DNA that tells the cell to make the PEG10 protein. And then also a piece of DNA that tells the cell to make a piece of messenger RNA that has the gene of interest, such as CRISPR-Cas9, but flanked by these packaging signals, the 5' and the 3' UTR sequences. And then finally, we put in a third piece of DNA that expresses a fusogen. And this is a protein that would come on the surface of these virus-like structures and then allows the virus-like structures to enter their recipient cells. So on the right side are electron microscopy images showing what these particles look like. And so cells naturally secrete particles called exosomes. And so that's what the negative images look like. So you can see that they're smaller and they have different sizes and shapes. But the ones that are derived from the PEG10 virus-like particles are very uniform in size. And they look very much like a virus, except they are made out of human protein. And so you can see that they have these round virus-like structure. And if you look at it carefully, you can see that they have these different layers. You have a lipid layer on the outside, and then you have these capsule-like structure on the inside. And so this shows that these are really very, very much virus-like things, except they're endogenous to the human body. So we try to see, can we use this to deliver a CRISPR-Cas9 to achieve gene editing in recipient cells? And so this is an experiment without very much optimization. We were able to package both the CRISPR-Cas9 gene as well as the guide RNA, and we can quite easily get fairly high levels of gene editing, and this is 40%. So this is already approaching a level where it's therapeutically relevant. So this is just one example of how we are developing gene therapy and genetic medicine technologies. And I just want to show this slide, which the plot on the right shows the number of genomes that have been sequenced over the past 2 decades. And you can see that the rate of accumulation for new genomes is growing faster and faster. And now we have tens of thousands or hundreds of thousands of different genomes. And many of these different genomes are from organisms that live in very diverse environments. And so inside of these genomes, there are many, many interesting biological systems that remain to be studied. CRISPR or these sort of virus-like proteins like PEG10 are just examples of what we can discover and mine from the natural diversity, and we can harness them to turn them into molecules that can improve human health. So we're very excited to continue this work, and just want to take a moment to acknowledge my collaborators, our financial support from different funding agencies and private foundations, and also if you have any questions, you can feel free to get in touch with me. Thank you very much. It's really a great pleasure to be a part of this panel.
Thank you very much, Feng. That was a great presentation, and we look forward to the later discussion. And now we're going to turn to Dr. Gao. So Dr. Caixia Gao is Principal Investigator at the Institute of Genetics and Developmental Biology, the Chinese Academy of Sciences in Beijing. Welcome.
Thank you very much, Dr. Johnson. Thank you for the invitation. Today, I'd like to share our recent story entitled Genome-edited Polymutal Resistance in Wheat Without Growth Penalties. Genome editing has become very exciting in crop breeding and agriculture. Genome editing itself provides a lot of new opportunity. We know crop improvement requires a constant creation and the use of new allelic variants. Here, I just want to introduce 4 major ways people perform plant breeding right now. Traditional crossbreeding has been used for thousands of years, in which people can take 2 different plants and cross them together and through many cycles of back crossing, resulting in a new variety. However, this is a very long and laborious process. It typically requires 10 years to create a new crop variety. Furthermore, it is limited that we can only introduce traits that are already present in the parent's plant. Mutation breeding has expanded the genetic variation by introducing random mutations throughout the genome of plants using chemical mutagens or physical irradiation. However, these mutations are random and non-specific, and generating and screening large numbers of mutants is very challenging. Very often, mutation breeding also needs back crossing. Transgenic breeding is also known as genetically modified plants. Transgenic breeding can introduce designed traits through the introduction of foreign genes into the genome of the plant. Foreign DNA is randomly inserted into plant genome. Although the GMO is efficient, it requires a lot of different screening to find a good event with durable gene expression. Furthermore, GMO is subjected to extensive governmental regulation around the world. Many countries today still do not allow to grow GMOs for use. Genome editing is very specific and targeted. We can even deliver our genome editing proteins directly into plant and allows us to directly edit the plant genome with no DNA incorporated into plant genome. So genome editing rapidly accelerates the process of crop breeding. Traditionally, we may take 10 years to breed a new crop, and now with genome editing, just in 2-3 years, we can create a new crop variety within a designed trait. Also, because genome editing allows us to edit the plant genome without any transgene, public acceptance is increased. It's much easier to allow a genome-edited crop into the society. A country like America, UK, and also China recently shared a new regulation on genome-edited agricultural crops. So it's exciting to see where genome editing will be in the future in agriculture. Today, I wanted to focus my story on wheat. Bright wheat is a stable crop that we all eat day by day. Bright wheat is hexaploid, meaning it has 3 separated genomes. Bright wheat genome is also very big, 17.1 gigabases, which is over 38 times larger than a rice genome. So wheat genome is very difficult for genetic manipulation. The main problem we wanted to address with our wheat genome editing is powdery disease. Powdery disease is one of the most destructive diseases affecting wheat worldwide. In this paragraph, we can see that in China, the epidemic area of wheat powdery per year is on average 7 million hectares of land. Serious yield losses can up to 40% in some places around the world. So it's a very important disease to address. Current methods to address powdery disease in the field rely upon chemical fungicide. However, chemical fungicide is harmful to both humans and the environment. So we wanted to develop a new method in which we can use genome editing to breed a new crop variety that is resistant to powdery. For powdery disease, there is a known susceptibility gene called the MLO gene. The MLO gene is a conserved gene across many plant species, such as barley, Arabidopsis, tomato, and pea. It has been previously shown that the knockout MLO gene in these 4 different plants, the plant was resistant to powdery. We decided to look at whether we can generate a new wheat plant immune to powdery using genome editing. Because wheat is a hexaploid, there are 3 homologs of MLO in wheat on chromosomes 4B, 4D, and 5S, respectively. In 2014, my group used talent genome editing to design a pair of talent that can simultaneously target all 3 copies of the MLO gene in wheat. We demonstrated that when we knock out all 3 copies of MLO gene in wheat, the wheat inhabits robust resistance to powdery disease. Unfortunately, although the MLO AABBDD mutant plants were highly resistant to powdery disease, they also inhibited early senescence and the defects in both plant height and green yield in comparison to wild-type plants, in which limited its application in agriculture. Fortunately, we also identified an interesting mutant. We named it as MLR32. Like the MLABBDD mutant, R32 exhibited robust resistance to powdery. Intriguingly, the R32 mutant displayed no growth penalties in terms of plant height and green yield when growing in the field. Since 2014, we spent nearly 8 years to uncover the biological basis behind the genome. We performed whole genome sequencing to precisely genotype our R32 mutant. We identified a 304 kilobase pair large deletion on chromosome 4B of R32. The red border of the deletion was located in the MLO-B1, and the left border terminated in an MLO-like gene we newly discovered and named MLO-X. Given the sequence similarity between the 2 genes, we speculated that the large deletion may have been due to a talent of target event. To investigate the effect of this large deletion, we performed RNA-seq of MLO, R32, and wild-type plant. we discovered that a gene named TMT3, which is located immediately upstream of the deletion, was markedly upregulated in R32 plant relative to wild-type plants. TMT3 is a tonoplast monosaccharide transporter. Its function is to transport monosaccharide from cytoplasm into vacuum. Previous study indicated that overexpressing TMT gene can increase seed yield in Arabidopsis. Assay for transposes accessible chromatin sequencing demonstrate that in wild-type plant, the transcription of TMT3B is repressed by the presence of the repressive histone mark H3K27 trimethylation and the inhibitory chromatin loop, whereas the large deletion in R32 abolishes this epigenetic repression and creates a permissive chromatin environment around TMT3B, thus resulting in its increased transcription. So through this study, we demonstrate a new wheat genotype, which we name R32, that inhibits robust disease resistance to powdery disease and is high yielding. The mutant relies upon 4 simultaneous multiplex edits in wheat plants. All 3 copies of the MLO gene are knocked out, and a large 304 kilobase pair deletion is generated in B genome. We wanted next to apply this discovery to use genome editing and breed new elite wheat variety. We use CRISPR-Cas9 DNA and RNP to develop new wheat germplasms harboring the R32 genotype. We used only a pair of single-guide RNAs to achieve simultaneous editing of all 3 MLO genes and the designed large deletion in the wheat genome B. We applied this method to more than 10 elite wheat varieties grown commercially and successfully generated transgene-free wheat crop with a designed genotype in only 2-3 months. So these findings highlight genome editing as a powerful strategy to rapidly and efficiently generate elite wheat germplasm with robust powdery resistance and enhanced crop growth and yields. I would like to thank every member of my lab, both past and present. I also like to thank our collaborators and our funding agencies. Thank you very much.
Thank you very much indeed. That was a very interesting talk, and shows the power and the speed with which gene editing can really change plants in interesting, important ways. We're now going to move on to Dr. Armand Séguin. And Dr. Séguin is a microbiologist and research scientist at Natural Resources Canada.
So good morning, everybody. Can I have the first slide, please? So I'm working with the Natural Resource Canada, with the Canadian Forest Service. On the second slide, I will tell you a little bit about biotech's story to introduce the potential of CRISPR technology in the forestry species. So that story starts with the spruce budworm, which is an insect defoliating spruce species in Canada and the US. You can see in the middle, there's budworm on the left, and the dead trees in the middle, the dead spruce trees. And one way back in the days to control that insect was spraying the biological insecticide with planes. You can see the planes with the BT tanks with the white ones that were really effective in killing the spruce budworm. On the next slide, this is another component that will be important for CRISPR technology, is really the tissue propagation, tree culture for propagation of trees through somatic embryogenesis. It's very efficient technology. So tree breeders have been collecting seeds of high value with trees with rapid growth and good characteristics. And essentially, we could put them into tissue culture and make lots of small plantlets that are totally viable. And so that will be, and put into the greenhouse for planting. And that will be the vehicle for the spread of the technology that I will talk more about. On the next slide, it's really the summarize of the story that I want to tell about the transgenic BT spruce that was done almost 20 years ago. So in the figures in the middle, it's really to put take the toxin, the gene encoding the toxin in the Bacillus thuringiensis, and introduce that specific gene into plant cell, which is really typical of the plant biotech industry. The corn BT and all the BT plants that have been created. That was possible back in the days because of a collaboration between the Rockefeller Foundation and a close colleague, Professor Armand Séguin at the University of Ottawa. So we were developing a technology for gene transformation in spruce, based essentially using like the gene gun or the gold particles with DNA that have been shooted to the cells in culture, the somatic embryogenesis cell system culture, or also now we're using agrobacterium for the transfer of the gene. So the idea was to have obtain a copy of the synthetic cry1AB genes from Bacillus that is able to express well in plant. That was tested in rice by Professor Althazar and under the Rockefeller Foundation. And on the next slide, you can see the small on the left or on the right, I don't know what to say. Well, the test tubes with small spruce trees that are transgenics. And you can easily see after the feeding trial, we just put like a larvae of the spruce budworm in each of those test tubes. And there's two remaining in the healthy trees in the middle. And those were the two different transgenic lines. So it was really effective to produce those, to have like transgenic trees, spruce trees, and producing that Bacillus thuringiensis toxin. So we decided to go out and plant those trees and do a field trial. And the long story short, on the right, you can see the transgenic trees on the top with the healthy needles or a healthy tree, and the one in the back at the bottom where we put essentially we put a budworm in those small bags and let it go for a week. And coming back after a week, all the foliage, all the needles from those trees have been defoliated. So on the next slide, I would like to move into more CRISPR technology and essentially the perspective and the challenge for tree gene editing in the context of carbon sequestration to focus more in a general context of the climate change. So as you can see on the diagram that I have, in yellow, we have the DNA transfer and propagation technology developed. On green, we already have expertise in the field trial and environmental assessment. But in blue, it's really the missing component to move fully into CRISPR technology with trees. And on the next slide, you can see that if you look at and it's almost like a green light for us at that time to prevent going into CRISPR technology. If you look at the size of the different living organism, the size of the genome, Arabidopsis being the smallest plant genome size, poplar in the middle is deciduous trees, but that was the working model for trees in our case. But if you look further right, I mean, spruce and most conifers like pine as well have used genomes. Roughly, we're talking like 20 gigabase size genome, and this is almost like 5 to 6 times the size of the human genome that was obtained in 2004. So we had to wait a little bit. So it's really around 2013, 2014 that we got access of conifer genomes. And on the next slide, I'll just tell you a little bit more about that. So there was like pine genomes, spruce genomes, and now because of the improvement of this DNA sequencing technology, we got like portal like 3 genes DB, where we got several genomes deciduous trees as well, like ash trees, spruce, silver fir, redwood genomes. So we're in business to tackle more problems using CRISPR technology. And that was essentially funded through big consortium in Europe, in North America, in Canada, in US, Genome Canada, for instance, to help us doing the basic groundwork for getting like annotated a full genome of huge size. On the next slide, I did my homework a little bit to see what was done in terms of CRISPR technology in forestry species. So since poplar genome was first available, essentially poplar is on top of the list with several papers on CRISPR technology, essentially for engineering wood properties in most cases. And the two first papers were published essentially in 2015, engineering essentially lignin pathways. And also many of those CRISPR technology in forestry species have been tested using the phytoindiesterase knockout to make the trees albino to remove chlorophyll. More recently, and it's really recent, I mean, there's three forest species that have been engineered, case study, eucalyptus, pine, and spruce. So essentially what they did in eucalyptus, they made sterile tree by overexpression of a leafy gene. And that was an interesting component. And essentially, as it was mentioned earlier, propagation of the GM trees could be a concern from the environment. So the fact that those trees are sterile, they don't set seeds, so they will stay local if planted for specific environmental or industrial use. Also, in terms of conifers, because as I mentioned, we had to wait to get clean genomes to do something. So there's two examples, one in pine, one in spruce, where they did the proof of concept of the fact that the technology is working. But it's going to take a little longer to get like a full set of a portfolio of projects like in poplar for conifers, because of still the size of the genome and the fact that the annotation of the genome needs to be perfect. Heterozygosity is problematic because you can identify some genes that you want to knock out, but you never know if you get another allele somewhere hiding because of the genome is not fully complete. On the next slide, just want to give you an example of targets for CRISPR technology, and it's plant pest resistance, for instance. So if we have healthier trees, we should eventually have a healthier forest that will be less susceptible to fire. So that was the premise when I started looking at the literature. And there's many cases like now we can engineer spruce budworm using CRISPR technology based not on the BT, but other genes that have been identified in spruce. Mountain pine beetle resistant against the mountain pine, resistant in pine for the mountain pine beetle is also possible using CRISPR technology because of the database and the knowledge that we have. We can also think of a resistance to fungal disease as well. But after reading a few papers, I hit the statement right in the middle saying that scientists continue to debate the effect of beetle killed trees might have on fire behavior. So, I mean, I think it's going a long road to make sure everybody's on the same pace. But intuitively, I think the fact that if we have healthier trees, we should get a healthier forest, less susceptible to fire. But let's not go into the details. On the next slide, it's kind of summarizing a little bit the different component of the talk. So now we got the field trial done, environmental assessment, and one component important is social acceptance. And I think it's kind of on top of the not the pyramid, but the different component of that. So the carbon sequestration side on the right is relatively straightforward in the sense that we know how wood is made, how cellulose, hemicellulose is made. So we can engineer that component to make it more safer in terms of paper production, for instance, by using less chemicals. The carbon sequestration is clear that the understanding how wood is made is important, but also an important component of the carbon sequestration is taking care of the soil. So the soil microbiome work and science is expanding very importantly recently. So we will need to take a more comprehensive approach with all the components. I mentioned forest health early on with the spruce budworm, the mountain pine beetle, and there's another component, another tree species is ash, which is on North America, susceptible to the emerald ash borer, a defoliating insect that is almost killing all the ash in North America. And in Europe, it's the ash dieback, which is a fungal disease. So that could be also another tree to tackle to help the mitigation of climate change. It's an urban tree as well, so it's really close to the people in the city, and the canopy provided by ashes is really important. On the next slide, and the last one. Okay, well, sorry. Well, it was more like a point to made where I saw on the Boyce Thompson Institute website that they were mentioning that gene editing can prevent eucalyptus from becoming invasive. And this is the leafy CRISPR project that I mentioned just before. So there's different perspective in seeing the value of CRISPR editing in trees. And you can read that. This is all what I essentially touched upon before, like root products, the economical values compared to the ecological and the social issues. I just want to thank my research team and the colleagues for the great work they accomplished. Thank you very much. Sorry for being over time.
Thank you very much. I think that was a very comprehensive overview of what might be possible with trees. And I think there's obviously some discussion to be had at the end of this presentation. So now we're going to turn to Dr. Peter Phillips, who is Distinguished Professor of Policy and Founding Director of the Center for the Study of Science and Innovation Policy at the Johnson Soyoma Graduate School of Public Policy at the University of Saskatchewan.
Thank you very much. It's a pleasure to be here today. In many ways, I'm going to key off and try and bring a little bit of context about the use of these technologies that Drs. Gao and Séguin have talked about. I'm an international political economist. I'm interested in how technology gets used. Will it change the productive capacity and the welfare of society? And I work with a group based at the Global Institute for Food Security in Saskatoon. Is it moving forward? There we go. I want to talk about three futures and then about three of the bigger challenges that have already been alluded to, but we have a little bit of evidence of how they're working in Canada. The three futures, much of what we've heard already is what I call endgame number one, which is that we apply these technologies in combination with all the data that makes them valuable in agriculture and forestry and other areas. And we are able to almost in an unlimited way, finally and quickly and cheaply deliver targeted traits for targeted uses for targeted markets. And this is an important part of the system. It's a big industry. It's a $60 billion industry globally. It's about 10% of the cost, but it's only about 10% of the cost of moving a product to market. And it's coming down. So that's the good news, is that this has a lot of prospects for advancing our productive capacity. A critical part of that, though, is will farmers adapt and adopt it? And part of this story is not just the seed and whatever you put in it using gene editing, but all the other ancillary tools that will allow them to make money. And the vision you hear from some of the more advanced companies and public actors there is that we can script agriculture. We can take the uncertainties and the diffusion or the errors out and lead to a cheaper and more profitable and more productive agricultural sector. And in the sort of the end game that some people tell me about is that they would like every farm to be interlinked with the research system, so that every 10 meter square block of cultivated land is a de facto field trial. And so you're getting continuous feedback, and that could be important. And this is a critical part. This is a huge part of making technologies work. It's about half the cost and about 30% of the effort we put in globally. The third story that none of us have alluded to is that people want to impose their interests on farming. And so there is a school of thought that says this will allow us to do just right foods. You'll get exactly what you want, when you want it, produced by who you want it, where you wanted it, and how you wanted it. And the mountain bike is the quintessential example. Nobody built it. It was a bunch of users who got together and said, if we could just tinker this and that, and all of a sudden you have a new product line. In the food space, it's entirely possible that consumers will get into a two-way conversation, that they'll start specifying, and that will complicate the way we deliver food in the world system today. So let's take these three visions, which are transformative to the way we feed the world today, and talk about some of the challenges, because they're not trivial. They're not insurmountable, but they won't resolve themselves without some effort. The first challenge, which is a problem we all have in the technology space, is we live in a field of dreams kind of world. We build these technologies and we say, if only I can build it and make it work and show the world that it'll work, they will beat a path to my doorway. The teams will come and play on my field, or as the earlier adage was, that if you build a better mousetrap, they'll beat a pathway to your door. The question is that true? How do we ensure that things that work get actually used by people in productive ways? And we've been out and sort of testing some of those questions, and the view is that these technologies, not unambiguously, but with a fair degree of support from a group of international experts, anticipate that it will drive our productive capacity in agriculture. So there's some positive elements, but you can see it's critically derived partly by not just the tool, but all the ancillary data that goes around it, the genomics, the farmer metadata, the phenotypic data that allows breeders to actually put these together in packets that are actionable in the commercial space. And so that some of these are more and less, but all of these are probably required to make the technology work. The difficulty we have with technology is that we assume that it's a rational, simple process for somebody to assess and use the technology. And it's everywhere from upstream in the lab to at the farm level to at the processor level to at the consumer level. And the presumption is that we tell them what's there, we persuade them, they make a quick decision, they implement it, they confirm it by use, and then they adopt. That's the model we all think is there. The reality is that particularly with new technologies where there aren't exemplars out there, people need to trial this. They need to really know that what they're buying will change their conditions materially. And that's not a simple process. And sometimes it's not possible to trial things without actually adopting them. So we get into a bit of a holdup there. The second problem that Dr. Séguin has already referred to is all these things that are beyond the transaction. How will others view this? What kind of opposition? What kind of locational effects are there? What kind of networks are required to either make it happen or might be barriers? And ultimately, what social license exists? But at the end of the day, the people who are bringing these things to market are doing a cost-benefit analysis. They're adding these things up. And it's entirely possible that there's a good technology that will have material effects that people will not use. And so quite often technologies get stranded. So we have to temper our enthusiasms a little bit and say that not everything will work. The challenge is going through this process, and it takes years. So the more we shrink the research part, the more we're going to be required to think about the adoption stage. The second one, which has been alluded to already, is what I call the alphabet soup of regulatory space. It's not just satisfying your national regulator, it's all the products that will be using these technologies will be internationally traded. So you have to find a way of managing access and use in multiple markets. And right now we've renationalized the regulation of the food system. And one of the results is that we get differing decisions. They come at different times, which slows up adoption. And sometimes they give different absolute decisions. The Europeans, for example, are leaning to treating GM gene-edited technologies as GM technologies, which will bifurcate the global market, which complicates adoption. And when we talked to our experts and we said, what significance will this have in the ag sector? And everybody was pretty enthusiastic about their country. They thought this would have material effects in their country fairly quickly, sometimes less than 5 years, but definitely in the 5 to 10 year period. Global markets similarly, but when we specifically asked about the EU, they said they don't see much change there. And so we're going to have that continuing pressure. And we know that the regulatory stage is about 38% of the cost and 50% of the time to market. And it's getting longer, not shorter, as we learn things. And so this new technology might speed up research, but it might actually slow down the regulatory space. So there's a challenge. And all these numbers are up significantly since these new technologies started to get developed. The third one is, will it deliver value? And part of that's making it findable, accessible, interoperable, and reusable, the data around the technologies. And then there's the ancillary benefit sharing questions that are inherent in all of these, because many of much of the knowledge we're using has traditional sources to it. So there's a collective benefit, authority to control, responsibility, and ethics mode. So we asked both personally and institutionally what people saw as barriers or challenges to their adoption and use of these technologies. And what we found is that about 75% of the people, the researchers, were motivated to share data to accelerate the use of gene editing, which is good. But there was no majority for any single piece of technology or data that they were willing to share. There were pluralities, good, strong pluralities of some, but weak in others. So there's going to be a tapestry of access to data that will make the technology work. That's the personal level. And that what barriers did they say were there? They said there were significant problems in just funding and paying for the access and use to their technology, and that there were uncertainties about what it would do to their ability to exploit their own technology. So there are real questions that people have that need to be responded to in terms of the policy space. There's also institutional barriers. Bottom line is there's shortage of skilled people, shortage of resources, shortage of investment, shortage of leadership and vision. So we're fixated on the technology, but we're not understanding that larger package of digital sequence information. It's going to be critical to make these things applied and used across the food space and the forestry space and into the health space more broadly. And there's some, but what they did say is that there are a whole bunch of incentives that would encourage them to move towards sharing. And we do many of them right now. The formal requirements for citation and acknowledgements, that the building of access and use in some of these databases. So there's prospects. It's not like we're without tools. We just need to generalize these tools. So what are my key messages? The new breeding systems undoubtedly provide a real opportunity to move the bar on food productivity and food distribution and food resilience, so that we're not as subject to ups and downs as we are with our conventional technologies. But adoption and use is going to be a lot slower than people are wishing for. And it's partly the economics, that the value proposition for individual breeders in Canada, for example, there's no public breeder using this to bring a technology to the market. They're using it for research tools, and then they re-engineer it back into conventional or transgenic technologies. The regulatory system is still a work in progress. Canada's only halfway to deciding how we'll deal with gene editing in the food space, and others are in similar ranges of degree. And the incentive structures at the individual and institutional level are mixed. So there's some thoughts for how these technologies that have great promise may have challenges before we get to full adoption and use. Thank you.
Thank you. Thank you very much. I think there are some challenging thoughts in that presentation, as well as really, going from the research to the actual adoption in the biotech and in the field. So it's going to be an ongoing challenge worldwide. Now, our final speaker is our moderator and our host today, Dr. Rodolphe Barrangou. He is also a Canada Gairdner International Award Laureate 2016, the same year as Feng. And that was his work in really defining the components of the CRISPR-Cas9 system in bacteria as a bacterial immune system. He is the T.R. Kleinhammer Distinguished Professor at North Carolina State University. Rodolphe, welcome.
Thank you, Janet, and good morning and good afternoon, everyone. It's my great pleasure to be here and actually be the last speaker of this very exciting, very timely, very relevant session. So without further ado, this is the business that we're in. We're really trying to turn science into products that are accepted by both consumers and regulators, as we just heard. And sadly, but also opportunistically, maybe science is the easy part, right? Thanks to people like Feng, thanks to people like Caixia, we have the knowledge and the understanding of the effectors of the tools, of the toolbox, and of its implementation in general, and also in food and agriculture, and eventually in forestry. That's great, but the challenge is that we have to address the bottleneck of acceptance and regulations. So where do we stand right now? I mean, just 10 years ago, Feng and his lab were providing the first ever proof of concept that CRISPR can be used to edit the human genome. What a momentous decade. And in just 10 years, a lot of the tools that Feng and others have developed have exploded. Over 200,000 dissemination to not-for-profit organizations throughout the world in over 100 countries. That's great. That's illustrative of the speed and scale at which this disruptive technology has been adopted. It's very competitive scientifically. That's why people like Feng, Caixia, and others are leading the edge and the field, right? Pushing the boundaries. And also there's geopolitical context. That's why we're here, right? Internationally. It's a global perspective panel series on genome editing technologies today for food and agriculture and forestry. And we see that the academic leaders in the world at the national level, of course, the US, of course, China, even Canada, almost in the top 5, are deploying those technologies. But importantly, we should all note that a lot of this is being driven by therapeutics, right? Like 95% of all this work is focusing on human therapeutics. And that's great because it's pushing advances, it's technological push, it's maturation of the technology and the tools and the toys and the toolbox, as we heard today from Feng. But I would argue that we have a prioritization conundrum. Because if you think of the number of people who will benefit from CRISPR-based therapies and cures in the next 10 years is relatively small relative to the number of people and consumers who can benefit from the full extended deployment of genome editing and breeding technologies for agriculture, for food, and for forestry. And I think as a gathering place, right, the Gairdner Foundation spirit to convene people and thought leaders, I think that's a conversation we need to have a little bit more of, is how do we prioritize and how do we strategically invest in and enhance and enable the deployment of valuable technologies for the betterment of humankind. Now, again, thanks to Feng and others, right, just 10 years in, we've gone a very, very long ways. If you go back to challenge number 1, as we just heard, patients have been dosed. It's clinically relevant, it's clinically efficacious. A lot more patients need to be dosed, a lot more diseases need to be addressed, and gene therapies need to be developed. And hopefully the delivery modalities that Feng and others are working on will change the world. But we need to turn that technology and deploy it more at speed and at scale in forestry. And that's why we need delivery modalities, virus and retroviruses, right? You can imagine how far we are on that front on the plant side. And then we need the tools in the toolbox to do that at speed and at scale, like Professor Gao illustrated. Now, despite the fact that 95% of all the CRISPR work is focusing on human applications, there's been a lot of work nonetheless on the deployment, the proof of concept across all kinds of species. We heard about crops, of course, fruits and vegetables, non-food edible crops, like think tobacco and cotton and hemp, and of course, trees and cellulose. And we've made a lot more progress, I think, than people know, people think, right? The non-browning mushroom, obviously livestock, obviously crops, for disease, for traits. And we're addressing endgame number 1 here, right? The deployment at speed and at scale of powerful technologies for food and agriculture. And it's just me saying it, it's the World Economic Forum saying it. And this 2 or 3 year old report predicted that by 2030, right, which is just like 7.5 years away, by 2030, maybe up to 15% of all the farms on planet Earth, 100 million farms will have planted a gene-edited seed. This is a challenge. This is an opportunity. Right? We may be on the cusp in a very relatively short time frame. Right? 10 years ago, we just had Crispr. 10 years from now, how far along will it be deployed on the continuum of food and ag? And of course, I would argue, and I'm working very hard on forestry, there are opportunities to impact even more people if we deploy that technology in the forest for decarbonization. But there are different kinds of challenges. We just heard about end game number 2, right? Scalability and deployment at speed and at scale at the farm level. And I can tell you, having gone to commercial forests in South America and throughout the world, the scalability of the forestry industry in terms of time and in terms of physical space is daunting, but it's an opportunity. And already there are people and companies and organizations, for profit and not for profit, working on using those breeding technologies that we just heard of from wheat, right? Just a few talks ago from Professor Gao, right now working in trees to alter lignin content, to lower lignin content, to make it more sustainable and amenable to processing, to increase the SG ratio, to increase the CL ratio, and of course, maintain height. And we need tools like genetics, as we heard. We need tools like machine learning. We need tools like a CRISPR toolbox, a delivery toolbox, and we need tools that enable us to bring it to the real world and scale that up and ensure that we don't just optimize the traits for processing and pulping, but also maintain the otherwise important integrity and phenotypic features of those trees. We want to maintain height, we want to maintain diameter, we want to maintain volume, but we also want to maintain health and longevity and environmental stewardship and the like. Not just address one trait, but address multiple traits if and when possible. And with those great potential and great technologies also come great responsibilities. Of course, we've learned that from the therapeutic world with ethics. And in 2022, we'll understand that accessibility and equity, that having access to those disruptive technologies and deploying them responsibly is very important. We've heard almost in every single talk about public relations and social acceptance, endgame number 3, as we just heard. We need to engage with the public. We need to have thought leaders like the people on this panel help engage the public, engage regulators, engage investors, engage politicians, so we can move this along. Again, regulatory path, challenge number 2. Caixia mentioned countries like the US, like the UK, like Japan, and like China that have been relatively proactive in leadership positions to move those regulatory frameworks ahead to ensure that we do so in a timely manner. And of course, there's technical solutions to that as well. Non-DNA, right, DNA-free, non-GMO, non-transgenic modalities to really, truly spark, to ignite, and to enable a sustainable agriculture and create that bioeconomical value that we just heard from economists, but also articulate why it matters in terms of sustainability and environmental gains for more enhanced, sustainable, energetically responsible, chemically responsible, sustainable agriculture. And that's why we gathered here today. That's why we're here. And that's why we're going to go straight into this Q&A here, because we have to collectively, the scientists, the technologists, the developers, industry, governments, academia, profits, not-for-profits, and society, we all need together to be able to have those conversations, to transcend those boundaries and all the challenges that we heard about in the last hour and 5 minutes, so we can get to the next stage. So at this point in time, I'm going to take off my presenter hat and put on my moderator hat and ask that the production team bring the panel together here. Again, personally, I will use this opportunity to thank, again, the Gairdner Foundation for convening us here today. And of course, Telus Health for their generous sponsorship and support. So let me start maybe with a short series of questions to each individual panelist. And maybe I'll go in order in which we spoke. So Feng, you've articulated very clearly, and you've done a lot over the past decade to drive the technology here. Why do you think that in just 10 years we've gone so far?
Thank you, Rodolphe. That's a great question. I think the reason that we have gone so quickly and so far is because things accelerate when you have a convergence of ideas and techniques. It starts to make doing things easier and easier, and that allows progress to be made much faster. Biology is really at a very exciting juncture. We have learned a lot about biological systems. There's a tremendous there's unimaginable amount that we still need to know, but the tools and some of the basic information is starting to turn biology into a more engineering-like discipline, where you can systematically start to answer a lot of questions. And I think that's probably a part of it.
Well, like that. So yeah. I'm going to jump off of that actually to go straight to Professor Gao here. So based on what we just heard from Feng, right? We now have the tools, the knowledge, the know-how, the technology at pace, right? To enable with velocity, right? Feng, to your point, the deployment of those technologies, in your case, in agriculture for wheat, as you articulated. So outside of the science, right? Caixia, what do you think has been a key driver of both the scientific and the regulatory progress that China has been able to really advance at a very impressive speed, right? China is a leader in the technology, but also, as you mentioned, a leader on the regulatory front. Why do you think you've been able, with your colleagues and compatriots, to advance the technology and also concurrently make regulatory progress?
Yeah, thank you, Rodolphe. That's a good question. I think that's the most important is, firstly, agriculture is very important in China. This food security is always the number one. So then, secondly, I think it's also because of technology. I also mentioned in my slides, if we compare GMO and genome-edited plants, there's a big difference. Genome-edited plants do not contain any foreign DNAs, which is much easier for public to accept it. So I think that's one thing. And the second thing is also because of the method. Actually, it's very simple. It's very easy. Currently, it's very easy to make precise edits. We can make they are not called a gene. We can make a precise base substitution. We can even precisely insert a large DNA insertion. So, and in this case, as I also mentioned, in China, in late January, the Chinese government released a new rule. Of course, still genome-edited plants is regulated under GMO, but the regulation, the rules are much more simpler than compared to GMO.
That's great. I love the proactive position and the strategic investment and commitment, right, for food security at the national level. So let me then jump off of that directly to Armand Séguin here, and then say, in light of what we just heard, right, the ability of a nation, the ability of a regulatory system, the ability of a scientific society to advance the tech and put in place frameworks that are enabling. You made a very articulate case on the challenges that lie ahead in forestry. And in some cases, some of those issues are exacerbated. So what do you think we can do, or what do you think we should emulate, right? And whether it's China, whether it's Japan, whether it's the UK, whether it's the US, right? We can make that a global conversation. But what do you think, Armand, would have to be done to truly convince or engage or discuss with the forestry stakeholders what we just heard from Feng and from Caixia?
And it's a good point, a very good question. Forests are natural organism, not domesticated. And this is one particularly touchy aspect with the public. People like to go into the forest and relax, to do camping. It's not the same, for instance, comparing to a maize field. You will not really meditate in the middle of a maize field, but you will most likely do it more into a natural forest. So the domestication is an important bottleneck for acceptation. That's why I mentioned a little bit ash, because ash is a true urban tree. Working with urban trees might connect more with the people because they don't want, having their trees or in the park being cut because of specific disease. But I've been struggling for that question for the past 15, 20 years. You do one thing, they say, well, the other side is not good. It's very difficult to find a path where you will not be really prone to big critics. But the domestication of trees is of concern by the people. Crop is different. We thought initially working on Christmas trees because there's a heavy load of chemicals to have like nice trees as we can buy them at the market and stuff like that. I don't have a clear answer to that. I'm not sure I will be able to see that in my lifetime, unfortunately. But I think I've sit on many committees and international panels where social acceptability. One last thing is in CRISPR's technology in crop, you can breed out one allele and get rid of, but you cannot. Breeding out spruce will take at least 12 to 15 years before you reach maturity. So breeding out the component of the CRISPR technology is not an option. So what technology we can introduce, So it's going to be like the weakest component that will be the problem of the whole acceptation with all the benefits.
I agree. So let me jump off of that again. I'm using the comments from one panelist here to go into the next, and I see Peter expecting the question here. So Canada has been, and we know that factually, a leader in the science, right? It's a top 10 country in publications and patents and the like, and innovation and startups, and so we know that, and ag and genome editing in particular. So what can we do for Canada to kind of stay ahead of the curve, not just in science, but also in technology acceptance, right, Peter? Like, what can Canada do systemically and strategically to ensure that within Armand's lifetime, we will be able to really deploy at speed and at scale in forestry and in agriculture and in food for the benefits of the Canadian people across regions and provinces and the rest of the world, right? What can Canada do? What should Canada do to address that?
Many of the things we've been doing in the past are the right things. The challenge is that too often governments, Canada's not alone in this, look inwards to aligning all their regulatory and social systems. And in a world of social media that slops over national boundaries and international NGOs and international business, it's not about a single country. So part of it is taking international leadership where we can. And in some product categories, we are international leaders. We work with other countries to help them to understand the technology, help them to adapt and adopt the technology in their own space, but also to understand that the positives and negatives of each choice that's made in the technology innovation space. And that's a critical factor. Too often, I think in the past, particularly in the GMO world, we locked into our particular vision of how the world should be, and then we tried to market it outside in Africa and Asia and Latin America and said, you need to buy our model of regulation and then you'll be happy. I think we need to be a little bit less prescriptive and more assisting. And if you go back in the modern technologies and go back into some of the early products that we were leaders in Canada, like canola, we spent, the reason it grew into being one of our biggest and best products in the Canadian agri-food space was that we spent a lot of time and energy working with the world so that they could see how it worked in their industrial space. They could see how it worked in their own agro-industrial context. And we could help them to identify the appropriate quality and safety concerns and address them. So we didn't let them make it up to their job to figure out how to make it work. And I think there's more that can be done there. It's getting harder because it's no longer a nationally controlled space. We're dealing with multinational companies and international research teams. So it gets more difficult, but it's still possible to take some leadership and put the pieces together that are on the table, but they're not coordinated in any way that leads to acceptance in a product space.
Yeah, I would agree. Again, I'm just going to make a comment here before I ask my next question. So I agree that it's more complicated and interwoven, but I will also point out that in today's world, the connectivity, right, the transnational organizations, people's ability to not be so nationalistic and really understand the context, right, at scale, dynamically for equity, for accessibility, for security and for sustainability, right, to address grand challenges as a collective, I think also provide opportunities to have a little bit of a more productive dialogue, right, but engagement, but we'll see that. So let me ask an open question to the panel here, and anybody can answer the question first and second and so on and so forth. So we've discussed much today about where we've been in the last 10 years. Again, thanks to Feng and people like Caixia have been able to advance that and deploy it at speed and at scale. So I would like to hear from the panelists, if you had to pick one thing that is going to be critical for the next 10 years, either a problem or a challenge or a technology or an event or a milestone that may occur, right? What is it that you're looking forward to that 10 years from now, when we have another Global Perspective Panel series and we look back on the first 20 years of genome editing, we'll be like, we were able to do this, and that's been a game changer. What would that be?
Okay, I can start. I think the development of effective and a universal method for delivering CRISPR-Cas reagents into plant cell is a challenging task.
I would agree.
Yeah, we just saw Armand's slides. Actually, we see when we wanted to edit a plant genome, conventional method relies on delivering a CRISPR reagent into plant genome. Typically, this CRISPR expression cassettes are delivered and expressed with cleave chromosome and then produce this site-specific DNA double-strand break, leading to this modification during DNA repair process. And then this, in plant cells, this introduced the CRISPR-Cas or DNA usually become integrated into plant chromosome. Then the plants need to undergo selfing or crossing to remove this CRISPR compound while maintaining this designed genome edit. But this process, of course, after selfing, after segregating this CRISPR expression cassette, this plant genome will not contain any foreign DNA. But in some cases, this process requires multiple generations, especially if we think about this tree, for example. This Armand just showed us, it takes many years. Therefore, I think to develop an efficient DNA-free genome editing approach by which mutant plants can regenerate it by transiently expressing this CRISPR-Cas9 DNA or RNA or RNP will be very important.
Very good point. Yeah.
Maybe I'll jump in there. While I think international regulation and getting the science fully articulated is critical, I think that too often in our conversation we ignore the breeder and the people who multiply the seeds out. We assume that if we make it available that they have the tools and the capacity to use them. And what I hear from breeders when we survey them is that they feel squeezed between all these uncertainties about whether it will be acceptable and appropriate in the industry, and people saying you got to use it. And that's grantors, that's policy people, that's industrial actors, and sometimes it's even farm groups. So I think the breeders need to be worked with in a more sustained way to address their concerns, because if they don't adopt the technology, it's never going to get used. They're the conduit, they're the choke point, and they're humans, and they have uncertainties, and they have extreme angst about, in Canada at least, about when and how it's appropriate to use the technology. And we've detected that angst, but we haven't got any immediate responses to it. But I think that will be part of what our grantors and our policy people and our institutions that support and sustain them will have to deal with. Because you can't just leave it to them to fix the problem that everybody else is presenting.
I think we might need also some winning stories, because as you mentioned, with the social media, news goes around the planet quite rapidly. I like to bring back the example of the eucalyptus, which is less a reduction of weediness of eucalyptus because of engineering a specific genes that prevent seed production. So I think this is maybe a bad example, but I mean, if we have more of those cases where there's clear advantage in engineering plants for specific traits, not necessarily for commercial aspect, but this is more difficult when there's no commercial to cover all the costs for regulation and things like that. So I think it needs to be more democratic in a way where, you know, small things, it could be just the change of a color of a plant or some stuff that as scientists we see this is not very useful, but I mean, it could really, I mean, the people can really be impressed by that and see the benefit and being used to it. Exactly what the level of scientific knowledge of the people in general about viruses, even the viruses because of the COVID situation. I mean, if you go back in time and ask the same question, the people will not provide the same answers just because they're more aware. So there might be like something with plants where, you know, there's that people don't understand and their fears that, and so by having really clear examples, winning stories might help. But which one? Wow, that's a big challenge.
Feng, please.
I think one thing that I think a lot about is education. I think it's really important that we continue to provide a robust training pipeline for new scientists, for kids today, get them excited about science and pushing the frontier. I think that is something that we need to do more to really, you know, help us continue to innovate and develop solutions for world's problems.
So that's perfect, Feng, and maybe it's a perfect opportunity here to set up our closing in a minute, because one of the missions of the Gairdner Foundation, right, is to inspire the next generation of scientists in Canada and throughout the world, and hopefully that kind of global perspective panel series will do that. And education is important. So in closing here, I'm gonna ask the panelists to provide an answer in 1 to 5 words, right? Answer the question as follows, right? In a short sentence or very even in a single word, right? CRISPR will change the world of sustainable food and agriculture because
Better understanding
to education. Yeah.
It links needs with opportunities.
Means and opportunities. Ooh, I like that. Caixia.
Because the technology is very precise and easy to use.
Feng.
Because we can engineer crops and other organisms to be more sustainable.
Great. Fantastic. So we're right on time. I thank our panelists for their time, their engagement, their wisdom, and their ability to challenge and be thought-provoking to our audience. I'm going to thank again the Gairdner Foundation for organizing and convening this, and of course Telis Health for their support, sponsorship, and enabling this to happen. So thank you all for your time. Thank you all for your consideration, and of course our attendees. And I look forward to future Gairdner Global Panel series and to provide perspective. Thank you very much to all.